Good afternoon, and welcome to the first afternoon session of the Canadian Genomics Summit. I hope you had a wonderful lunch. My name is Cami Ryan, and I am Senior Business Partner for Industry Affairs and Sustainability for Bear Crop Science here in Canada. I'm also a member of Genome Canada's Board of Directors and am Chair of the Science and Industry Advisory Committee. I'm really delighted to introduce this panel to you today. How we explore genomics and can enhance the resilience of Canada's agri-food, environmental, and climate change systems. I just took a bit of time today to go to Webster's or Oxford or whatever and get the definition of resilience. And in the dictionary, it's defined as the capacity to withstand or recover quickly from difficulties. And I guess in this context, in Canada, it means a little bit of toughness. And it is also the ability to bounce back into shape. So we're navigating a period full of complexities and challenges. The landscape we face today includes various factors from trade dynamics to weather patterns and biodiversity shifts. And all of this really shapes the world we live in, the world we work in, and the world we interact with 24/7. In Canada, we're experiencing weather events like droughts, heat waves, and flooding that impact our safety, our agricultural health, and of course, food security. Additionally, biodiversity loss is of concern, and this affects ecosystems across the country and all over the world. But if we refer back to what resilience means, how it's defined, we need to more than just bounce back from these challenges. We need to seek out, support, engage in opportunities that enable us to not only bounce back, but grow and innovate. We need to think and do beyond the notion of just resiliency. We need to make the pie bigger. Genomics, as we all know, offers promising solutions to many of these pressing issues we face. For instance, genomic surveillance has become a vital tool for detecting and managing emerging plant diseases. And by monitoring pathogen genomes, researchers can effectively alert us to potential threats. And that helps us to manage these diseases and safeguard our crops. Genomics research is also paving the way for the development of climate-smart crops. At the University of Saskatchewan, for example, scientists are working on creating resilient crop varieties that require less synthetic nitrogen. This innovation not only helps farmers save on their costs, their input costs, but also reduces fertilizer-related emissions. And moreover, the impact of wildfires, as we've seen both in Canada and most recently in California, highlights the importance of resilience in our ecosystems. Research at the University of Alberta is uncovering why some trees are more resilient to wildfires, drought, and disease. This knowledge can enable us to breed trees that are better equipped for local conditions. These are just a few examples of how genomics is contributing to our resilience in Canada. Genomics is our path for navigating these many challenges, and genomics is a path forward for Canada to bounce back and go beyond. Over the next hour, we'll delve into even more examples of ways genomic research enhances Canada's food security and productivity, and how that also influences the rest of the world, and how genomics strengthens our supply chains, protecting biodiversity, and helping us to address the issues associated with climate change. To lead this discussion, I invite Dana McCauley, CEO of Canadian Food Innovation Network, to the stage, and I also invite our panelists to take their seats.
It's interesting, the Cami started to talk about resilience, because just moments before, Christine, who I'll introduce properly in a few moments. And I were talking about our panel and about how, wow, the topics that we heard earlier today, cancer and like, these, responding to these huge challenges. When you think about food and agriculture, it's hard for that, we felt our topic to be as relevant and as emotionally impactful. But when you think about resilience, you can't rise to those amazingly difficult challenges and solve those very complex science problems unless Maslow's hierarchy of needs has basically been taken care of. You've had a warm, safe, dry place to sleep, you have a full stomach, you're not worried about where your next meal is coming from. So I think when we move into our panel, I'd love for you to just keep that in mind, that contextually, looking after the environment, biodiversity, food systems, and food supplies is really, it's just so foundational that I think sometimes we all take it for granted. I've segued way off my notes, and but thank you, Cami, because that was a really inspiring way to start. So as Cami mentioned, I run the Canadian Food Innovation Network. I am not a scientist in any way, but I do spend a lot of time listening to scientists and trying to understand what is possible in the realm of technology and IP-driven solutions, because the purpose of CFIN, we're an economic development group, we want to help every food and beverage business in Canada thrive, help us to grow our GDP, create great jobs, and most often they can do that by adopting technologies or using new ingredients or new techniques to deal with their waste in ways that will help them to grow their business and often as well have a really positive impact on societal problems. We invested a lot early on. CFIN's going to be 4 years old on April 1st, which is both hard to believe and like, wow, feels like a long time as well. And in our first couple of years, we invested quite heavily in funding projects that de-risk technologies in the bio space. We partnered with Ontario Genomics, who's here today, to do a program where we both put in money so that industry could participate and academia could participate with less commitment. We really wanted to encourage that. And then we also put about $2 million into some other projects. And we did that because I like to, and I always recommend to anybody who's in the funding space, like to approach it as a portfolio type of an opportunity. What will hit commercialization tomorrow and have an immediate impact, but also how do we think about the future and make sure that Canada will be resilient in the future. And I put the genomics work that we've done into that latter category. And I have to say the investment is paying off. We dropped a first of its kind report on Tuesday called the Food Tech Ecosystem Report. And we have discovered through the research that we did that Canada has, 3 domains where we are leaders internationally in the food technology space. And one of them is biofood tech, specifically biofood tech enabled functional foods. So I think that is pretty fantastic and very hopeful for us as well when we think about how the future might favor Canada in new forms of trade. I should have started out by thanking Canada for having me here today. I'm expecting to learn a lot as your moderator, and I want to just kind of take some of the things that they told me when we first started to talk about this panel and just get you a little bit excited about some of the things that Genome Canada is already invested in. So creating more nutritional value for our food sources, crops that are more resilient, and taking measures to protect biodiversity are, of course, all things in the realm of Genome Canada and their various partners across the country. A specific example is in Quebec, there's a genotyping of Canadian sheep project, which is developing much more, resilient and hardier strains of sheep so that we don't have to rely on imports. That's quite an exciting project. Genomics is also creating ways to help nature adapt to climate change. The Innovative Genomics Institute has been using CRISPR to enhance the ability of plants to both capture and store carbon. And these are investments that are just going to make Canada much more competitive, much more resilient, and ready kind of for anything. Enough of me. I have this amazing panel. Unfortunately, one of our panelists has been caught up in the weather and won't be able to join us, but if anyone sees Callie come in through the door, just send her up. I'll share my mic with her. We'll get her on board. But truthfully, each one of these people on the stage could probably fill a full hour themselves. Christine Bays, Chair of the Department of Animal Biosciences at the University of Guelph. Thank you, Christine, for being here. Michael Luhais, who is the Vice President of Genetics and R&D at Cemex. And Lauren Ryberg, who is a botanist from the University of British Columbia. So we are going to sit down now, and I'm going to do a bit of a lightning round with the panelists. But before we do that, you guys get to participate. We're going to do a poll. So I actually said, one of the conditions, I said to Catalina was like, well, I'm only going to moderate if you'll give everybody like an Oscars-worthy loot bag at the end. And she said no. So we have, that wasn't in the budget. So we've compromised, and instead you're going to get 2 word clouds to take home. The first one is going to appear up on the screen in a moment. You'll get to scan a QR code, and then in one word, because it's a word cloud, put in what you think the greatest challenge facing genomics is for the next 25 years. We want to have something to put up in 25 years so that we can celebrate how we've conquered all of these challenges. And then in a little while, we'll come back with our next word cloud. Right now, I'm going to sit down. Whoa, or maybe I'll just ruin the furniture, who knows? Everyone's, I'm sure, regretting having me here at this point. So, guys, you have all been actively working in the genomics space for quite some time. Maybe 25 years is not a milestone that you've reached, but regardless, I'd love to start out just giving you an opportunity to give a slightly more detailed introduction of yourself, and then just to highlight the thing that in the last 25 years in your space that you work in that has been sort of the biggest, most exciting thing to celebrate in genomics. So, Christine, you want to start?
Sure thing. First of all, thank you for giving the opportunity to be here with Dana, and at least I didn't fall on my chair. Just saying, but I probably will fall when I come off the stage. Yeah, so my name is Christine Bays. I'm currently the acting Associate Dean for External Relations. I got bumped out of my chair position to take over that. Recently, I hold a Canada Research Chair in Livestock Genomics, and I am in the Department of Animal Biosciences within the Ontario Agricultural College at the University of Guelph. I love cows. I love turkeys. I love chickens. I love livestock. Really a lot. I don't know if that comes across like I want it to, but I really do. And I think it's really exciting right now that we're in a time when we look back at the last 25 years. I was born, Filippo, by the way, so people make fun of me sometimes, but I was born. And when I think about the introduction of genomic selection, which I think is probably the biggest thing that fundamentally changed how we select livestock today. Genomic selection was implemented in Canada 2009, 2010. By 2015, LactaNet, which is an organization that we work very closely with at the University of Guelph, they had about 1 million genotypes in their database for cows. By last year, that number increased to about 8 million, and it is increasing constantly since then. So this is I think, pretty impressive. Genomic selection basically uses SNP-chip technology to select our animals based on a palette of different traits. And the best thing about it is that whereas 25 years ago, our focus was almost exclusively on production traits, things like milk production in cows or breast meat yields in birds, we can now improve the health and the welfare of our livestock unimaginably. We are now able to select for traits that are expensive, that are really difficult to measure, and I think that's probably the most fantastic thing that could happen for the welfare and the health of a really important source of high-quality protein for Canadian diets.
That's
Ultimately,
awesome. I'm going to ask you to stop
Sure,
there.
I'm done.
That is pretty
I'm really
amazing.
excited.
We'll expand in other answers. Michael, what's been like, what are you celebrating?
Well, I'll start there. And I'll tag team a little with Christine. Christine mentioned genomic selection, and when I was asked that question of what was the biggest accomplishment in the last 25 years, to me it was simple. It was the advent of the SNP chip. So genotyping on microarrays changed my world. One of the things that, and just maybe a little background on me.
Yeah, I'll just pause too. Does everybody know what a SNP chip is? Is that a common?
A SNP is a single nucleotide polymorphism. It's kind of a low-grade marker, but there's a lot of them. And what really helped was being able to put tens of thousands of these SNP markers on a single chip. So you can run a single sample of DNA across this microarray and come up with 50,000, 100,000 data points. Why I say that changed my world is because 25 years ago, I was working for a company, a Fortune 500 ag company in the States based in St. Louis, you might be able to guess. And we were genotyping, we thought we were, cutting edge. A big part of my budget went to microsatellite marker genotyping. You might remember the LICOR machines. These are gel-based electrophoresis, very slow. A big accomplishment was getting the cost down to $1 per data point. And now, I think it's more than 3,000-fold cheaper. So we can get 50 to 100,000 data points from one chip for $20 to $25. So it's an amazing, what it has done is it's democratized genomic selection. So anyone, whether you're a university researcher or whether you're an ag company like we are, I work in the cattle breeding industry. We sell semen, embryos to 80 plus countries around the world. And what this has allowed us to do is to be a leader in a very competitive world. So we'll talk more about that later.
That's awesome. Thank you. That's really interesting. Now, Lauren, I did a horrible job. I just said he's a botanist. So, please take a moment. I know it's way deeper than that. Tell us a little bit about your work and then what you would love to celebrate.
I am a botanist.
Yes, but it was very, very high level intro. I apologize.
I'm a plant evolutionary biologist, and so, Christine, I love sunflowers and cannabis and carrots. But I think cows are friendly. I just don't want to eat them. So I study though the wild relatives of crops, and I also run a small breeding program for vegetables for the organic growing sector. And so I kind of span two research areas, biodiversity and agriculture. And so I thought of what was an innovation that kind of spanned both areas, and I decided to focus on one called landscape genomics. And this is an area that's really been pioneered in Canada by Sally Aitken, who's here in the audience, with the Adapt Tree projects. And the way it works is that, and this was invented by evolutionary biologists, that you can identify genetic variants in natural populations that adapt those natural populations to their local environments. And the way this is done is simply by by looking for associations between features of the landscape, such as temperature, precipitation, soil fertility, and genetic variance. And this has been a remarkably powerful approach. In biodiversity, for example, it's been used to predict the responses of natural populations to climate change. It's also been used to identify maladapted populations to current or future climates and suggest solutions. In the sort of plant breeding sector, by doing landscape genomics with crop-wild relatives and with land races, it's a very simple way of identifying alleles under extremely complex traits. It can be very hard to phenotype, like Christine was saying, some of these physiological traits, but you don't have to phenotype them. You can just use landscape genomics to find alleles associated, let's say, with with fertility stress, low nutrient soils, or with high temperatures, or with drought. And then you can move these into breeding populations and create climate resilient varieties with very little phenotyping and very little expensive phenotyping. So on the crop side, it hasn't been used that much yet, but it is the future, I think, and will really play a big role in adapting agriculture to climate change.
That's awesome. Thank you. I learned a lot there. So now I just want to dig in a little deeper with each of you, give you a little more time to explore some ideas. Christine, really interested in your thoughts on the role of genomics in addressing climate change, specifically, adaptation strategies and just the things that I know in your work are really trying to be proactive and mitigate what could happen in the future. So, floor is yours for a few minutes.
Absolutely. I think there's two ways of looking at this. We have adaptation and mitigation. As many of you know, livestock do contribute to the greenhouse gas emissions produced in Canada. Dairy cattle in particular are about 1.2% of our greenhouse gas emissions. If you add beef cattle to that, we're probably at about 4.6%. Canada is the first country in the world to have developed a national genomic evaluation for methane emissions of dairy cattle. That's amazing. I don't know last night someone talked about celebration, and this morning celebration as well. I think we should celebrate that. We're a leader. And companies like CEMEX can use that for export, etc. It's really important that we contribute to mitigation of the impacts and the footprint of livestock. Mm-hmm. That was allowed, or that was made possible through probably 10 years of really intense collaboration between industry partners, other stakeholders, farmers, universities, and funding from organizations like Genome Canada. It wouldn't be possible without that. And I feel honored to sit here amongst doctors and people who work at sick kids, and it's always a little intimidating, but I feel like it's very important too. We've got a wicked problem with climate change. Our next large-scale project aims to reduce the CO2 equivalence of dairy cattle by 55% before 2050. That's a reduction of 6.5 megatons of CO2, which is a lot. And it might only be 1.2%, and half of that is only 0.6%. But that's a big contribution, and we have to all do exactly the things that we can do. Now, with regards to mitigation, that's kind of mitigating, but what about adaptation? Our cows and our livestock still have to live in a warming climate. We're breeding across the board. We have the capabilities now of collecting more than 100 traits are being currently collected on dairy cattle. We're working on beef cattle. Plants and animals together are being selected for resiliency traits. Cattle are being selected for heat tolerance. We have identified a gene, the royal we, not me, just to be clear. There's a slick gene that's been identified that is prominent in heat-tolerant cattle. This becomes incredibly important if you think about food security, not only within our borders, but outside of our borders as well. Drought-tolerant crops, et cetera, et cetera. The list goes on, and the applications of genomic technologies in agriculture they've been interwoven into the practices that are used every day.
And that's so interesting, and you did mention working with industry to accomplish some of these successes. But I know from being somebody who's always sort of bridged, industry and academia, as a broker of sorts, both when I worked at University of Guelph and now in this role, that sometimes industry still has some frustrations and doesn't necessarily know kind of how to engage, et cetera. And so, Michael, you obviously are very successful at working with academics and knowing how to translate this foundational science into, commercial value propositions. What can improve in that relationship to ensure that genomic innovations reach farmers, reach other countries? Like, what have you learned?
Well, I feel very fortunate to be in the place that we're at. We have a great partnership between our partners in the academic world. University of Guelph has been, in lockstep with us in terms of what to work on. What are the traits that farmers care about? What are the traits that society cares about? We can provide the demand, but the ideas and the skill set comes from our academic institutions. When you're at a party, alcohol may be a social lubricant. When you're in research, the lubricant may be funding. Funding has a way to bring people together. And so Genome Canada has
Cash
done...
talks.
Wonderful. I really, since coming back to Canada 8 years ago, I've seen a research environment that has done a great job of supporting not just universities, but industry as well. And so that has lubricated this relationship that we've had. And, one of the things that we have to tip our hat to are these partnerships where one group has stored data. For example, I was talking earlier with a colleague about mid-infrared spectra data. Never would have thought that would have been important to climate change. But Lactanet decided to keep this data, to store it over several years. And a University of Guelph researcher asked the question, hey, could we predict methane, which is very expensive, very difficult to measure, could we predict that using mid-infrared spectral data? And the answer was
Eureka.
70% correlation.
That's amazing.
90% correlation at the genetic level. So that was the thing, that was the eureka moment that allowed us to be first in the world to develop methane evaluations.
2 quick follow-ups before I turn to Lauren. One, you mentioned coming back to Canada and being heartened by the way these relationships were working. So I'd love to know if there's a difference between the 2 countries. And secondly, money, of course, is always helpful. But I would imagine that you also bring really valuable insight into the industry's problems and into the farmers' problems to researchers as well. Am I right there?
Well, I'm a farm kid.
Yeah.
I grew up on a farm. My first job was in the cattle industry, and I always felt when I went back to academia that I had an unfair advantage, knowing what the problems were, what were farmers talking about. Farmers are logical, cogent decision makers, and they will vote with their pocketbooks. They're making big financial decisions every day. So things like health of their animals was really important to them. Production, profitability, of course, that's very important. Talking about methane and what can we do better, right now, farmers, when you ask about methane, they'll say, okay, that's probably a good thing to do, but why should I select for that? It doesn't help
Well,
me.
this is it. It's not a business problem for them, right? It's a
not
societal
right.
problem.
This is the first
Yeah.
trait. As important as it is, the value accrues off the farm, not on the farm.
Yeah.
So what can we do better? Well, we have to make that link with our supply chain partners, with dairy processors, dairy retailers. They want to bring down their Scope 3 emissions. We can help them do it. Farmers need to be incentivized to do it.
Yeah, you need to layer the value for them, right? Like there's got to be a business value as well. Yeah, thank you. That's interesting. And then just quickly, do you see that Canada and the U.S. are quite different in the way industry works with academia, or not?
Well, there is more of a zero-sum mentality in the U.S. Now, it's much bigger. It's very aggressive. I will say I enjoyed my 19 years in the States very much. Don't judge all Americans by one orange individual. But there is a mentality of risk-taking that we don't have. And if there's one thing I would want to change is, how can we lose some of that risk-averse mentality that we have in Canada? We always play it safe. We're afraid to go bankrupt. So many successful people in the States go bankrupt
Interesting.
before they have great success.
All right. Well, that's a great topic to dig into another day. Lauren, you talked about this landscape approach, and you touched on the value to supporting biodiversity of that approach, and of course climate change and conserving land and forests and oceans and all those things are so interconnected. Could you just dig in a little bit more about what the work that you're doing, the types of projects and the outcomes that you anticipate that leveraging genomics will help to enhance biodiversity protection?
Yeah, I wasn't. I was going to talk about sort of overall genomic support for biodiversity, the biodiversity sector, and I guess the biggest one was mentioned this morning, is the ability of genomics to discover, identify, and delimit biodiversity, species, ecotypes, races, and so on and so forth. And this is an area where Canada has led with the barcoding project by Paul Hébert, the other Paul Hébert, down at the University of Guelph. And this has been this was supported by Genome Canada, and it's had a huge impact, because if you're going to be spending a lot of money protecting ecological communities or species or preserves, you really want to know what's there. And we don't have enough taxonomists to identify all these things, and often many of the species are cryptic. That is, there's no phenotypic diagnostic feature for them. You don't know until you look at the sequences. And it hasn't just been barcoding. There's also been these eDNA approaches, environmental DNA approaches, that have been hugely helpful in terms of quantifying the amount of biodiversity in a particular area, particularly for microorganisms that we haven't been able to do before. And then, yes, the other really important contribution, I think another one, is through the use of genomic approaches to identify populations that are at risk due to genetic or genomic reasons. And what I mean by this, populations that have lost their genetic diversity, or populations that are suffering inbreeding depression. Often it's the same thing, but not always. And populations that are maladapted or are predicted to be maladapted in the future. And genomic approaches can not only identify these populations and the particular risks that they and the risks to them, they also can suggest solutions. For example, genomic approaches can identify the best source populations to restore the diversity in a species, or to restore genetic health in an inbred population, and also to do assisted migration. Again, this is what Sally Aitken has led with forest trees, where you try to match the genotypes of the seeds you plant to the climate of the future. And so these are some of the, I'll probably stop there.
No, that's really interesting, though. I'd love to just ask you a little bit more specific. So Sally's work with trees is really interesting. What are some of the other types of plants that are, getting a lot of attention, and that you think we should be maybe some that maybe there are some that we should give more attention to? Like what are the vulnerable areas?
Yeah. So, well, I'm involved in a project funded by Genome BC to Greg Owens at UVic, who works on kelp. And one of the things that we discovered is that, so the thought is that kelp is suffering mainly because of anthropogenic activities. This could be warmer water, and it is, and also because sea otters aren't around to eat the sea urchins that are eating the kelp. But what we found in that project was that in some cases, particularly in some of the, in the Puget Sound and some of the interior waterways, these kelp were incredibly inbred. And the reason that they weren't healthy is because they had all these long strings of of the genome that had no genetic variation, and so they suffered very strongly from genetic load. So there's a pretty easy solution there.
Just go from bay to bay with some kelp, right?
Yeah, you bring some sea kelp, you bring some kelp in and from another location, hopefully fairly close by, and you promote some, you know, they shouldn't be mating with themselves or with their close relatives.
This is a PG panel. Okay, but yeah, no, that's a really interesting example. Thank you for that.
That is something that plants can do, is they can do it with themselves many cases. And so that's worse than mating with your brother or sister or your cousin, okay? Don't do it with yourself.
No. Okay, you heard it here. Maybe we'll get t-shirts, I'm not sure. Let's pause for a moment to put up the next poll. So we asked you about challenges, and I think there's some really interesting things there. I'm not surprised that funding is right in the middle, big blue word there. But now, you've heard some great stuff on this panel. Earlier today, you heard other amazing things, and each of you, I'm sure, is well suited to identify opportunities in your own fields and in your own communities. What, in one word, is the greatest opportunity for genomics when it comes to supporting our agriculture and natural science sectors? And let's see what we come up with there. And maybe this word cloud will be something that some of you will tack up on your whiteboards in your office and use to muse upon when you're thinking about what's next for your lab or your company. All right. So that everyone gets an opportunity to have one word, but I'd love to give each of you 2 minutes to think about, in your field and what you're doing, what is the big opportunity that you would like policymakers, you'd like funding bodies, you'd like journalists to be talking about. And I want you to, have a moment to have a really great answer, so I won't pick on anyone. I'll let you guys volunteer for who'd like to go first. Christine, you look ready.
I talk too much. I'm sorry.
No, you don't.
No, I'm excited. There's so many things to be excited about, and there's so many opportunities right now. The implication and integration of artificial intelligence, artificial machine learning technologies is going to be fantastic. Precision agriculture is going to be fantastic. Microbiomics is going to be fantastic, but none of those things are fantastic unless you have the people trained to be able to integrate them and weave them into application. So I'm excited about the ability to do that, and I would be excited about education if I felt like that was going to be really well funded in the future, but we're struggling there a little bit in Ontario and in Canada generally. But I do think that this is a very, very exciting time because Genome Canada, all of the people here have worked so hard to bring us this far. We have reference genomes. We have the ability to work with them. We have technologies that we're waiting to be integrated. I spoke earlier and we talked a little bit about collaboration earlier this morning. The ability of industry partners, funding agencies, academics, and even farmers to work together is going to be, that's going to bring us into the future of food production. Genome Canada has a scientific and industry advisory committee. They're looking out, and I feel really good about that they're looking at beyond our borders, about what else can we be learning from other countries, how can we collaborate with other countries. Our projects that we run at Guelph have multiple countries involved. So this interdisciplinary ability to work is good. We need to keep training people. We need to have highly qualified personnel, and that's going to be the future, and I think it's fantastic.
The research that we did in the report I referenced earlier that, highlighted biotech foods as one of these domains where Canada shows leadership. One of the reasons that our team cited for that ability and that go-to-market leadership was absolutely the pool of talent and of scientists who know how to use data and apply data science to solving problems. So yeah, you're right. That's a big opportunity for Canada is to continue to invest in that, because right now we're looking pretty good. But yeah, you've got to. It's like exercise, right? You've got to keep doing it to maintain. Yeah, good point. You want to add anything to that?
Yeah, I mean, I think that we can, if we were to invest somewhere, I think research projects are fantastic to invest in. We need almost a larger strategy of how to fix our university system as a whole as well, to keep going in order to maintain that spot. We're struggling with PALs, with the inclusion of new international students, bringing them to Canada. Those are things that I would ask any policymakers here to talk with afterwards.
Yeah, no, that's great. Thank you. I appreciate that. Lauren, what about you? What's the big opportunity in botany for the next 25 years?
I don't think it's just botany, but one of the things that as an evolutionary biologist we do is that we are very good at finding, as I mentioned, genetic variants that are involved in adaptation to the natural environment. And we're also very good at finding genes where there's a signature of natural selection that's been going on for a period of time, or you can see that a particular variant has swept across the genome, across a particular species. We call it a selective sweep. So we can see all these signatures. And in some cases, we have knowledge sort of vaguely that they're involved in adaptation to drier environments or something. But the frustrating part is we actually don't know what these things really do. And what we want to know, and I think there's a big opportunity for this with machine learning, as Christine mentioned, but also with gene editing and other functional tools, is that we have the ability, I think, with combining those two methods together to predict not only what genes do, but also what the particular variants do, the particular change in the protein or the particular change in the regulatory sequence. And so it's this predictive future that excites me maybe the most, because right now one of the things our field suffers from are lists of candidate genes. We hate lists of candidate genes. You don't have time to functionally validate all of them. And I look at the human literature with some envy, because you actually know a lot about what the disease-resistant variants we do. We want to see this across wild plants and animals and fungi and so on and so forth, is to have the ability to look at a genome and know what the different variants actually do.
So it sounds like you've got a lot of great info, but now it's learning how to apply it and interpret it and do something
well, there's a
more with it.
There's a lot of information that still needs to be gotten. In a sense, we know that a particular variant is important, has been under selection, but we actually don't know what it does. And that will, certainly will aid breeding programs if you actually know what the particular variant you're targeting does. And so we need a lot more, we need improvements in machine learning using the data we have.
Yeah.
For example, we use information from humans, in plants. But we also need a lot more functional studies of, there are so many genes in plants that aren't found in animals. And even if you compare two different individuals of a plant, like two different sunflowers, they may differ in their gene content by 30%. In other words, there are 30% of the genes in one plant aren't present in the other one. We want to know what those do.
Yeah.
So, and if you look
Interesting.
across the entire sort of population of sunflowers, you might find only 5% of the genome is shared by everyone. A lot of this will be the genes themselves, but there's going to be a core set of maybe 50% of the genes that's shared by everyone, and the rest are all variable.
So it's kind of overwhelming for someone like me to hear you talk, because then I think about just, I live out in the country, so I have quite a lot of space around me, but I just think about, you know, the many, many kinds of plants that are even existing on my little piece of land. And it's a staggering thing to get to the level of knowledge that you're talking about.
Yeah,
It's like sweeping the beach or something.
It's not as bad as it seems, because with machine learning, we should be able to.
It's not
we
so bad.
We should be able to make, you know, if we have sequence data, we should be able to make predictions. But yes, every time we get a new genome, there may be some additional genes that we have never seen before.
Interesting.
And that's going to continue for a while until we achieve saturation, which I don't know how long it's going to take. In certain species, like Arabidopsis, we probably are achieving saturation, but not in sunflowers.
All right. Well, that's very, very interesting. So, Michael, when it comes to, you know, your work, it's quite focused on you know, breeding, you know, healthy, resilient animals, and I'm sure that there is a big opportunity there. But you might want to speak a little more broadly, because you have a lot of perspective, I think, to offer from other parts of your career too.
Yeah, I've heard it said we're living in the post-genomic era. I don't feel that way. I feel like we're still living in the genomic era.
Yeah, it sounds like it.
It used to be the limiting factor were genotypes. And now I think we all agree in our field that phenotype is king. So that's what our limiting factor is having access to good, high quality, large enough reference populations where you have a genotype and a phenotype and hopefully some pedigree to go with it. You can do some amazing things. One of the things, and on the theme of being resilient, you've all heard about H5N1 circulating in the cattle population. This is a big concern.
Just today, there was an
someone
announcement that it is a new variant.
Yeah, in one of the milk silos, in one of the milk processing companies, there was a new variant that was responsible for infecting a human in the milk supply. Okay, so this is a concern. One of the questions that came forward, again from one of our research partners at the university, asked us, hey, you have a trait, it's high immune response. We call it immunity plus. What we've done is we created a reference population where we measured the immune response of animals to a disease challenge, or say a vaccine. We were able to measure the antibody-mediated immune response and the cellular-mediated immune response. Together, we packaged that as immunity plus. And one of our partners asked, well, how does that help with bird flu? You know, is there any benefit? And so we went to some of our customer partners, 6 farms in California that were infected with bird flu over the past year. They also happen to be genotyping partners, so we offer a genotyping service. So we already had their genotypes. We went to their health records and we looked, okay, was there an impact? So this hasn't been published yet, but what we did find was a statistically significant difference in the number of treatments that were required for those animals to bounce back. We're just looking at this, so it could have an impact on mortality and morbidity, which we have to study yet. But you know, isn't this a great thing when you can go back to a trait that was discovered through genomic projects and use it to actually help farmers mitigate and select for cows that are more resilient to these challenges.
Which is so important, because it's not just an economic problem to solve, it's obviously a huge food security and human health problem to solve as well. So yeah, that's an excellent one. We do have a few minutes. We can open it up for questions. If there are questions among the audience, we can take them, or I can just kind of freestyle. Oh, Nancy's got her hand up, so I'll move up here so I can get a good view. All right.
I have two, but I'll just ask one, and if there's more. I feel like, Dana, you touched on the relationship, but I feel like there's more to say on there. So I just wanted to see if there's anything more in the relationship between industry and academia. How did you handle IP as an example and things like that too? So I think you were very gentle in your responses, but I wonder if you can just be a little bit more on some of the stickier areas, how that relationship made it work, because it's beyond money.
Sure. So would you like industry or academia to start with their answer? Okay. Well, all right. So Lauren, did you have any comments on that? Because you actually didn't get to participate in that part of the conversation earlier.
Yeah, I've worked with a large number of different companies, and I find that it depends so much on the particular industry you're involved with. I found in the sunflower industry, the companies I created a consortium, a genomics consortium of the various seed companies that work in sunflower production. And it was a great thing because it brought the whole community together. So it was a public-private consortium. And we basically agreed that we would do pre-competitive research, and then they could use any information that came out to add IP to it if they wanted. And it worked out really, really well. And so we didn't have any problems. But I've also worked with cannabis companies, and it's a much more secretive and a challenging community. And so it seems like so much depends on the history and of the communities. There's much more secrecy in that community.
Would you say that that secrecy is related to the fact that I would imagine the sunflower people are fairly well established, ongoing businesses, whereas the cannabis companies are trying to seek funding from venture capitalists, et cetera, so they want to, have something very proprietary that's not public knowledge?
I think it comes from the history of the fact that it's been illegal for so many years, so everything's been
the illicit shadow.
Yeah, so everything's been secret for a long, long time, and so we tried to develop a consortium in cannabis, and it just wouldn't work. And so from my perspective, but yes, it's very much the history of, I think as cannabis companies become more secure, and they'll realize, but it's also a little bit different too. Cannabis companies are really competing with each other. And in sunflower, the seed companies are, sunflower is competing with other oil seeds rather than with sunflower per se. And so it's kind of a different competitive situation.
It's a scale issue.
And I think it makes a big difference on the attitude toward IP. A lot of the big sunflower companies, yeah, they're the same ones that also work on maize or soybean or so forth, and they have a much more nuanced view of what IP is useful and when it's not. And so I think as the industry matures, we'll get a more, so my relationships with, I don't have any complaints. So the culture was so different.
That's really interesting. So Michael, from an industry perspective, I know from when I have worked in academia or in academic structures, I was never an academic, but and now, when I was working on creating the funding programs for CFIN, for instance, we mostly have academics participate as paid service providers because we were worried about exactly what Nancy was talking about, getting into these IP issues. We do require all the partners to have an IP strategy that's documented and signed, et cetera. Industry seems to like that approach. VPRs like it because they get their overhead, so I was trying to be a bit of a peacemaker there. From your industry perspective, does it ever limit you from working with a university if you can't get that agreement nailed?
Well, it's funny to hear Lauren talk about it from the university side, and from industry side, we found that there were big differences between working with one university tech transfer office and the next. And there's also differences between researchers within the university. University of Guelph has been very understanding of the need to protect IP and very reasonable in terms of what that's worth and what it costs to actually commercialize something. Some universities have a very naive approach to what something's worth. They don't think about the cost of bringing it to market, where most products fail.
Right,
is
in that commercialization, that TRL 2 to 7 space.
Exactly, the valley of death, you know, it's just so many things die. And then, you know, within universities, some researchers really understand what's important to industry. They understand that in a competitive world, you need proprietary traits, something that's going to set you apart from your competitors. There are some traits that are table stakes across the industry, and there's others that set you apart. When I mentioned Immunity Plus, that was something that we held proprietary because that was really important to set ourselves apart. And we were able to establish IP protection for that work. Part of the IP protection is patents, but some of it is data, proprietary data. Those are things that can help industry. Now, one of the worst things I've seen is an academic who says, I just want my research to get out there. I just want to make it free. And it ends up having the opposite effect, because now there's no value. There's no value to one company paying to get through the valley of death and pay for that commercialization, because, you know,
There's no first mover.
there's no first mover advantage.
Yeah. Interesting. Christine, you were scribbling some notes. Either you were doing your shopping list or you really had something big to say on this.
I was drawing daisies. No, I was drawing sunflowers, actually. No, it's an interesting conversation. The experiences have ranged between excellent and awful.
Yeah.
Excellent.
Kind of like dating.
Right? Kind of like dating, right? Totally. No, I think at the heart of it, as an academic, I am an academic, and I will stick to my academic freedom, which somehow in these discussions still flows within all of the industry collaborations that we have.
Yeah.
Yet at the same time, we recently offered a number of organizations to actually sequence some of their animals, and they said, no, thank you.
Interesting.
Whereas other collaborators said, absolutely, we want to be involved, we want to understand. That was on a project, speaking of genetic diversity, regarding detrimental homozygous regions, so detrimental sort of inbred regions of the genome. I think that's very important. So we went ahead and collaborated with the organization that was going to, you know, allow us to do this. It's an important question, Nancy. Sometimes you win, sometimes you lose, and sometimes you don't get to do anything at all. But somewhere, if you have the right people and the right conversations, as it always comes back to people, you can still move ahead. And I think that's a hopeful message. You just have to kind of navigate it very, very carefully.
Excellent. Well, we are almost at time. It's, for me, been a really great conversation. I think takeaways that I will be musing on over the coming days are just the staggering, you know, breadth and depth of what needs to be done to be in a post-genomic phase, as well as, you know, just feeling really hopeful that there are people who are so engaged and curious and passionate about helping to ensure that we have a resilient food system and a resilient response to climate change, and you know, ensuring biodiversity. We've really crammed an awful lot of scope into a very short amount of time, and I would encourage those of you who are you know, foodies and curious to yeah, watch this this lane, because we didn't even get into talking about everything that's happening in cellular food and the creation of new ingredients. There's a massive conversation to have there as well. Thank you to all of you for participating in the word cloud. I look forward to getting my copies so that I can kind of study them side by side. I hope that was interesting for you. We will be taking a 15-minute break, and that'll give us all a chance to, get ready for the next great panel. But before we do that, I'm just going to ask you to please put your hands together for these three amazing speakers.